Okay, so now we want to talk about gene regulation. So now that we know how genes are transcribed and how they're translated eventually into proteins, all of this gene expression that we've been talking about in the previous unit are all regulated, right? Cells have the ability to control that expression of their genes. And so in prokaryotic cells, they're often controlled in response to environmental signals, which we'll talk about more in this slide. And in the next slide, there's an example that I want to share about how in eukaryotic cells, gene expression, whether a gene is transcribed and then eventually translated, are controlled often to determine and to differentiate one cell type from another, right? So the difference between a liver cell and a kidney cell is different genes are being expressed in those two different types of cells. And certainly different genes are expressed during different times of development in multicellular organisms. So that's what this unit is about. How are genes regulated? How do genes get turned on and turned off? And what controls that gene expression process? So as I just said, prokaryotes they're single-celled organisms, so their gene expression is often controlled in response to some environmental signals. And so in this slide here, we have an E. coli cell, and there in the box number 1, lactose, which is a sugar. Let's say that lactose becomes available in the environment. So, I don't know, the cell finds itself in an environment that's rich in lactose. So the cell then has the ability to change its gene expression in response to that changed environment. And so what is a cell going to need to do in order to utilize that new sugar source? So lactose is a new sugar source now that's available to them. Well, first of all, they need to get the lactose into the cell. And so some proteins, the one indicated here called lactose permease, allows for the lactose to get into the cell so that it can be utilized. There's another enzyme called beta-galactosidase that gets synthesized so that the lactose can get broken down and then the cells can actually use it. And so these genes then, we might say, get turned on in response to lactose in the environment. Before lactose is in the environment, none of these proteins or very low levels of lactose permease and beta-galactosidase would be there. But as soon as lactose is in the environment, the cell senses that and then turns on the expression of these genes that are going to be necessary for the cell to utilize that new sugar source. And so this is a common way of gene regulation in prokaryotic cells. And we're going to talk about a number of examples of those. So gene regulation in E. coli is often in response to an environmental signal, as shown here in this slide. So let's walk through this slide. So gene regulation in eukaryotes, as I said before, determines when certain genes are on or off, determines what type of cell actually gets developed in a multicellular organism. We know that at certain stages of development, there are certain genes that are on early in development, and then they're turned off in adulthood. And that's what this table here is showing. And I don't want you to get all caught up in the details. I'm just showing you this as an example of what I'm talking about. So this is a human life cycle, right, from fertilization through the embryo, fetus, and into adult stage. And we know there's a protein called hemoglobin. Some of you are familiar with it. Hemoglobin is the protein that carries oxygen. Certainly we know when we are in utero, the way we obtain our oxygen is quite different than the way we obtain it as adults, right? In utero, it's coming through the placenta of the mother, and then, of course, in adults, you breathe it through your lungs. Well, there are a number of proteins that are changed. Their regulation or their expression is altered during development. And so if we just look at that column labeled embryo and go down to the bottom, you see all these Greek letters. So there's a whole bunch of different globin genes, alpha, beta, delta, I think gamma and epsilon. So there's a whole bunch of these different genes that are part of hemoglobin. And what you can see during the embryonic stage, there are the two at the bottom of the list are the ones that are on, or in other words, those are the two globins proteins that are being expressed when we are in the embryonic stage. Then as we move towards the fetal stage, as the embryo develops, we now see that those two that were on during the embryonic stage are now off, and some other globin genes are being turned on. And then when we look to the adult stage, we see it's the top two, the alpha and the beta globin genes that are on, and all the others are off. And so there's a whole slew of mechanisms. And then that's the case for your entire adult life. Those three genes that are listed on the bottom of that bottom row, they are off. They're off for the entirety of your adult life. And you only needed those genes to be expressed during your early development. So all of that regulation, when and how genes get turned on and off through development, and in eukaryotes, it can be a fairly complicated process. So this is just one example of what I'm talking about here. So at what stages in gene expression are genes regulated? Well, we've on the left-hand side of this diagram, you see a bacterial or a prokaryotic cell, and on the right-hand side, you see a eukaryotic cell. And the thickness of the red arrows is indicative of how much control there is at each level. So we know, how do we control how much protein ultimately is there in a cell and how much protein is actually functional? Well, you could regulate transcription, right? So if the gene never gets transcribed into messenger RNA, then the rest of this doesn't happen either. Or if the gene gets transcribed at a very, very high level, then we would end up with lots of functional protein. So transcriptional regulation in both prokaryotes and eukaryotes is shown here. And transcriptional regulation is the primary way that genes are regulated. Transcriptional regulation is the main way that we regulate gene expression. And so we're going to talk primarily about that. But there are other points along this pathway where gene expression can be regulated. You could also regulate it in prokaryotic cells. You could regulate at the level of translation. Right? Well, if the messenger RNA does not get translated into protein, that also affects the amount of functional protein that's there. And then we can, in addition, we can modify those proteins post-translationally and that then result in functional proteins. And remember, when we were talking about gene expression, there's an additional step in eukaryotes that's not in prokaryotes, and that's that RNA processing step. Remember the capping, the tailing, the splicing? Well, that's an additional step where gene regulation or gene expression can be controlled. But for the most part, it is most common that gene expression is regulated at the transcriptional level, whether you're talking about prokaryotes or eukaryotes. So I want to start by talking about some specific examples of gene regulation in prokaryotes. They're fairly simple examples, but it really illustrates how, when you start to layer different mechanisms of regulation on top of each other, how this can get fairly complicated pretty quickly. So let's start by looking at gene regulation in prokaryotic cells and talk about it sort of generally. So if we're looking at the red here is the gene, right? This is the DNA. And we know that there are some important parts surrounding genes. We talked about this when we were talking about transcription. Remember, there's a promoter, which is always near the structural gene. And that promoter is important for regulating or controlling the binding of the RNA polymerase and allowing transcription to occur. Well, we can kind of boil this down to two primary categories. So we can talk about negative control or repression of gene expression, or in this case, we're talking about repression of transcription. We're regulating transcription in a negative way or repressing it. And ultimately what that does, when you're looking in this panel here on the left, is decreases the amount of transcription, maybe even shuts it off completely. So negative control or repression is carried out by proteins called repressors. And you see one of those here in green. And so what a repressor does is it physically binds to the DNA, to the gene, near the promoter. And because it's sort of physically sitting there, it blocks the ability of RNA polymerase to come in and transcribe. So when there are repressors bound to the DNA, you get little or no transcription of the gene. So that's negative control or repression. And on the other side, the other panel here, we see the opposite of that. You can also control gene expression at the level of transcription through positive control or activation. And activation leads to increased transcription or, more transcription. And this positive control or activation is carried out by proteins that we call activators. And so what activators do is they somehow interact with the gene, with the DNA, and they enhance or facilitate the ability of the RNA polymerase to come in and start synthesizing the RNA. So repressors, negative control, block the RNA polymerase. Activators, positive control, somehow enhance or facilitate the binding of the RNA polymerase to the DNA. And so these are kind of the two sides to the coin that we're going to talk about. We're going to talk about repressors and activators, primarily in prokaryotic gene regulation in the examples that we're going to talk about. But before we start talking specifically about their examples, there's one other player in this that I want to introduce you to. So whether or not a repressor binds to DNA or whether or not an activator binds to DNA is sometimes related to what we call effector molecules. Effector molecules are usually small and they physically bind to repressors, and this is an example of an effector binding to a repressor in this case, but there are also some effector molecules that bind to activators. And so when these effector molecules bind to either repressors or activators, they change the three-dimensional structure of those proteins. So in this example, what we're looking at is a repressor that is bound to the DNA, and it blocks RNA polymerase, and so you get no transcription in this case. Well, if an effector molecule, which is in the right side panel in blue, is present, it can physically bind to the repressor, and when it does that, it changes the three-dimensional structure of the repressor, the green blob, so that it no longer binds to DNA with high affinity. That molecular interaction between the repressor and the DNA is lost. And so as soon as that repressor is released from the DNA, the promoter is no longer blocked, the RNA polymerase can come in and turn on transcription. So these effector molecules influence the ability of the repressor, in this case, to bind DNA. So they're you can kind of think of them as sort of an on-off switch. So whether the effector molecule is present or not has an influence on the binding of the repressor to the DNA. And again, there are effector molecules that can do the same thing with activators. When they bind to activators, they change the three-dimensional structure so that it affects their ability to bind to DNA. So we've got activators, repressors, and effectors. So now I want to talk about some specific examples of how we'll look at a specific example of repression or negative control in prokaryotes. And we're going to first talk about something called the TRP operon. Okay, this slide shows some of the details of what we call the TRP operon, or the tryptophan operon. So tryptophan is an amino acid, and cells require the amino acid to live and survive. And so in bacterial cells or prokaryotic cells, operons are clusters of different bacterial genes that are all controlled by the same promoter. And in lab this week, we're talking about a different type of operon, and that is the arabinose operon. And the idea is similar here. So the TRP operon, again, is an example of negative control or repression. So on the panel labeled A here, we have our TRP operon. There's 5 different genes in that operon, TRP E, D, C, B, and A. And all of those enzymes are important for the synthesis of tryptophan. So when they're transcribed into messenger RNA, one messenger RNA ultimately then gets translated into the 5 different enzymes that are important for synthesizing tryptophan. And we also have a repressor. Again, this is an example of negative control. So we have a repressor that's shown there called the TRP repressor. So if a cell needs to, if these genes are on, it's only in a situation where the cell needs to synthesize its own tryptophan, right? If a cell finds itself in an environment with low tryptophan, then it needs to make its own. So transcription of this operon occurs under conditions of low tryptophan, or in other words, the genes of this operon are on under low tryptophan. And in panel B here, we see the same operon, but what it shows you the regulation that happens when cells are responding to their external environment in situations where the tryptophan levels are high. All right, so let's just look at each of these panels now in more detail. So we have our promoter in blue, we have that's labeled P, we have an operator in green, we have 5 different genes in the operon, we have a repressor, and then that large purple blob is the RNA polymerase. So in panel A, when tryptophan levels are low, remember, these genes are only on in that situation because that's the only time that cells need to synthesize their own tryptophan. And when this is on, there is no effector molecule present. Remember previously we talked about what effector molecules do. They bind, in this case, to a repressor and change its 3-dimensional structure. Well, in the absence or low tryptophan, there is no effector, and so the 3-dimensional structure of the TRP repressor does not allow it to bind to the promoter. So the promoter is totally accessible to the RNA polymerase, and transcription occurs. And the messenger RNA is made, and all of these enzymes then are there for the cell to use to synthesize tryptophan, since they find themselves in a low tryptophan environment. But what happens then when the cell finds itself in a high tryptophan environment? Well, tryptophan is the effector molecule. So if there's high tryptophan in the environment, it gets transported into the cell, and that effector molecule, or tryptophan itself, binds to the repressor, changes its 3-dimensional structure. So now that repressor can physically associate with the operator, and you can see in panel B there, it then blocks the ability of the RNA polymerase to move down that DNA and synthesize the messenger RNA. So we see repression under high tryptophan levels because the effector molecule tryptophan is present. So no transcription in that case, which is exactly what you would expect. If the cell has a lot of tryptophan in its environment, it doesn't need to make its own. So this is a classic example of negative control or repression. We have low tryptophan, no repression, because the TRP repressor is not binding, so we get transcription and translation of those enzymes. And then in panel B, the tryptophan itself is the effector molecule, binds to the repressor, changes its 3-dimensional structure that allows it then to physically associate with the operator so that the polymerase is blocked and it can't transcribe those genes that are normally involved in synthesis of tryptophan. And the cell doesn't need tryptophan under high tryptophan conditions, so that makes sense. So that's one really straightforward, simple example of negative control or repression. So now let's look at this same idea, but at a different operon. This one has a couple of other levels of control, and this is the LAC operon, or the lactose operon. And in this particular operon, again, you see we've got a LAC promoter that's labeled there in blue. We have another operator. And in this operon, there are actually 3 genes. And they are called LAC-Z, LAC-Y, and LAC-A. And they are all controlled by the same promoter. And when they get transcribed, you have a messenger RNA that contains the sequence for all 3 of those genes. And then those are translated into 3 different proteins. The protein encoded by LAC-Z, LAC-Y, and LAC-A. So same idea that we were talking about in the previous slide, but this is a different operon. And the other thing that I want you to note, in addition to the lac promoter and the operator at the front of this gene, we also have a kind of purple region there called the cap site. And we'll come back and talk about that in a little bit as well. So what does the lac operon do? Well, these three enzymes that it encodes, the LAC-Z, LAC-Y, and LAC-A, are involved in the metabolism or the utilization of lactose. Remember that very first slide I showed when cells find themselves in the lactose-rich environment. These are some of the genes that are turned on so that cells can use lactose. So let's look a little more specifically at that in the next slide. So again, when lactose is present in the environment, the lac promoter then is turned on that allows the cell to metabolize the lactose. So what are those genes? Well, one of them encodes the lactose permease. One of them encodes the beta-galactosidase that we talked about. And what I want you to pay attention to in this slide is this one component that's there on the far right that's called allolactose. So allolactose is sort of an intermediate in the breakdown of lactose, and that's going to become important in just a minute here. But so we've got the lac operon, important for the utilization or the breakdown of the sugar lactose, and there's a number of genes in the lac operon that are important for that metabolism of that sugar. And allolactose is part of that breakdown of lactose. And so let's now look at the lac operon, because it's an example of an operon that is controlled by both negative control or a repressor, and it's also controlled in a positive way by an activator. So we're going to break that down into its two component parts. So let's first look at the negative control of the lac operon, again involving a repressor. So there's two different states that a cell might find itself, right? When lactose is absent or lactose levels are very, very low. As I showed in that very first slide from this set of slides, you don't need all those proteins to metabolize lactose if there is no lactose around. And LAC-Z, LAC-Y, and LAC-A in this operon are the proteins that are important for metabolizing lactose. It can be shut off when lactose is absent. So when lactose is absent or low, this is what happens with the negative control. So again, we have a repressor. In this case, it's called the lac repressor. And that repressor, when lactose is absent, that repressor binds to the operator. And just as we've talked about many times before, it blocks the RNA polymerase from transcribing this operon. So the repressor binds to the operator in the absence of lactose, and you get no transcription of this operon. But now let's say that the cell finds itself in a very high lactose environment. So now we need all of those proteins that are important to synthesize or to metabolize and utilize this lactose sugar. So remember, we're talking about effector molecules, and remember a couple slides ago I said pay attention to that intermediate in the breakdown of lactose, which is called allolactose. So in this case, it's allolactose that's the effector molecule. And when allolactose is present, clearly that's when lactose is present, and allolactose then binds to the repressor, changes its three-dimensional structure. So now that repressor no longer binds to the operator, and transcription of the lac operon occurs. So we have the effector binding to the repressor no longer binds DNA, and it's now the DNA is accessible by the RNA polymerase, and we get transcription of the messenger RNA, ultimately leading to the synthesis of those proteins that are important for the utilization, the metabolism, and the breakdown of lactose. All right, so one of the four possibilities is that the cell finds itself in an environment where the lactose levels are high and the glucose levels are high. Remember that I said the cells always prefer glucose. So when glucose, this tripoperon is regulated by glucose levels in addition to being regulated by lactose levels. So when glucose levels are high and lactose levels are high, first if we look at the repressor, remember allolactose is the effector, and if lactose levels are high, then allolactose is present. It binds to the repressor, changing its three-dimensional structure so that it no longer can bind to the operator and the polymerase is not blocked. But at the same time that that's happening, the cap protein is there, and remember, high glucose levels inhibit the production of cyclic AMP. So with no cyclic AMP, the activator does not bind to the cap site, and so we do not have the high affinity binding of the polymerase. So while transcription might not be completely shut off, transcription is quite low because there's no activation. So in this case, when glucose level and lactose levels and glucose levels are both high, we get no repression, but we also get no activation. So transcription levels are really quite low of the lac operon in this case. All right, so that's one possibility. What's the next possibility? Another possibility is, of course, when lactose levels are high, but glucose levels are low. So this means the cell is finding itself in an environment where the only available sugar source is lactose. So this would logically be the case where it wants to utilize or metabolize the lactose in the environment. So what's happening here? Again, lactose levels are high, just like they were in the previous slide. So allolactose is being made, binds to the lac repressor, and so the lac repressor is inactive. In other words, it does not block the RNA polymerase. So we have no repression when lactose levels are high. But what's happening in the case of low glucose levels? Well, low glucose levels lead to the synthesis of cyclic AMP. That cyclic AMP binds to the cap protein. The cap protein then changes its three-dimensional structure, binds to the cap site, bending that DNA and really making that gene highly accessible to the RNA polymerase. So we get a very high rate of transcription of the genes in the lac operon, which is exactly what you would expect and want if you were a bacterial cell. My only sugar source is lactose, so I need to turn on these genes that are going to allow me to take that lactose into the cell, break it down, and utilize it. Okay, so this would result in high levels of transcription of the lac operon. Well, what's another possibility? Well, the cell could also find itself in a situation where lactose levels are actually low, but glucose levels are high. And if lactose levels are low, remember we don't want to turn on these genes that are in the lac operon that are going to allow us to metabolize lactose if there isn't very much lactose around. So in this particular case, there is no effector, there's no allolactose. Right? You need lactose metabolism to form the allolactose. So there's no allolactose, so the repressor binds to the operator and blocks the RNA polymerase from transcribing the lac operon, the genes of the lac operon. So that's the repression side of the story. But again, remember we're talking about glucose levels being high here. When glucose levels are high, cyclic AMP synthesis is inhibited. So the cyclic AMP effector is not here either. So we have no effector binding to the repressor. We have no effector molecule binding to the activator or the cap protein. And so cap does not bind the cap site, and we get complete repression and a very low rate of transcription here, which is exactly what you would expect because the cell is not going to synthesize these enzymes that are important for utilizing lactose if there isn't very much lactose around. And then there's one more situation. So this is the situation, again, where lactose levels are low, but glucose levels are also low. And certainly, again, when lactose levels are low, we don't need these enzymes that are important for metabolizing lactose. So again, no lactose, no allolactose effector molecule. The repressor binds to the operator and blocks the polymerase from being able to transcribe these genes. And in this case, because glucose levels are low, our cyclic AMP effector molecule will be there. And so it'll bind to the cap site. But because that repressor is blocking the RNA polymerase, that activation doesn't matter. So again, we're still going to get a very low rate of transcription under these circumstances, which is exactly what you would expect in situations where the lactose levels are low. So in this slide, basically we've summarized all the last 4 slides. I've put it all together here in one slide. So again, remember that the only time the genes of the lac operon need to be transcribed is when lactose levels in the environment are high. Okay, so whenever we see lactose levels low, repression, right? And you see that in the bottom 2 panels. And then when we look at the top 2 panels, the other condition here is the only time that lactose needs to be utilized is when it's the only sugar source available. So in A, we actually have 2 sugar sources available, right? Lactose is high, so there is no repression. But glucose is high too, so we're going to utilize glucose instead. So there's very little transcription here. So the only of these 4 instances where this operon is on is when lactose is high and there's no other glucose sugar source for the cell to use. And so repression is off or inactive, but activation is on. And that's the only instance shown here in B where we would get transcription of the lac operon. So as an exercise in helping you understand this, I basically put the 4 different conditions here in this table. I would encourage you, take a picture of this, draw it out yourself, and continue to go back to the previous slides we just went through and ask yourself these questions. In the situation where lactose is high and glucose is high, is the lac repressor on or off? Is the cap activator on or off? And what turns that lac repressor on or off? Is it repressing? Is it blocking the RNA polymerase if it's off? Is the cap activator on? If it is on, or what is turning that activator on? Is the effector molecule present or not? And then overall, by saying the question, is the lac operon on or off? In other words, is it being transcribed or not? So this is, there's a number of really good questions too on the worksheet for this unit, but I would encourage you to kind of work through that and draw something similar to this to help you, really think about how this all gets put together. And I guess I'll just leave you before we move on to talk a little bit about eukaryotic gene regulation. I hope that you can start to appreciate how complicated it can get pretty quickly. So you have both negative control and positive control in response to 2 different things, lactose and glucose, happening here at this one single operon. So you have a way, the cell has a kind of a nuanced way to respond to its environment. And when you start talking about gene regulation in eukaryotes, this gets layers upon layers of complexity can be put in here to really have some very tight, specific control over gene expression. Okay, so I want to switch gears and talk a little bit about gene regulation now in eukaryotic cells. We could do an entire semester, and there are such things, on eukaryotic gene regulation. There's so many players involved and so many ways that this works. So I'm oversimplifying things and just sort of giving you sort of the backbone or an outline of how this works. And I've shown you this slide before, but it's just to remind you, just as in prokaryotes, the primary step at which gene expression is regulated is at the transcription level. We're going to briefly touch on the RNA processing, the translation, and the post-translation mechanisms too, but I'm just going to briefly mention them. But so what we really are talking about when we talk about gene regulation is almost exclusively transcriptional regulation. So a couple of things before we dive into this a little bit more. So when we were talking about prokaryotic gene regulation, all of the proteins that were binding to the DNA were binding at that promoter. And you can imagine that there's a limit to how much regulation you can have because there's only so many proteins that can bind in that one specific area of the DNA. So that method of regulation is limited. And so eukaryotes have developed some other ways to do that. And then we also need to remember that eukaryotic genes are not organized in operons. Genes in eukaryotes generally, each gene is controlled by a single promoter. So they're not regulated in clusters like we saw before. So that gives you a little bit more fine tuning, right? You don't have 5 different genes that are all either on or off. You can sort of regulate each one of them separately. And eukaryotic genes are regulated by many, many proteins. And there's this thing called combinatorial control. So there's lots of different ways, and it's not all at the gene sequence. And so we're going to, again, lightly touch on some of those. But before we do that, let's look a little bit at what the eukaryotic promoter region looks like and talk a little bit about how the proteins are organized at the promoter, the proteins that are important for regulating transcription. What kinds of things do they bind to at the eukaryotic promoters? So here's a really simple diagram of a eukaryotic promoter. And the structural gene is shown there, so that's the actual coding sequence. And instead of having operators and promoters like we were talking about in prokaryotes, you have this sequence called the core promoter. And that core promoter is composed primarily of 2 different components. One of them is the transcriptional start site, or that the place where transcription begins. And then another one, and you see it shown here, 25 about nucleotides if we go to the left. That's called the TATA box, or the TATA box. And it's called that because the sequence there is usually some series of T and A. And that TATA sequence is really important for helping the polymerase find that transcriptional start site. And the core promoter, these sequences here that are right next to the structural gene, are really important for maintaining just sort of that low level, basic level of transcription. But of course, if we want to regulate gene expression, we have to have the ability to turn that dial up and turn that dial down. So the core promoter is important for just maintaining kind of that low level of transcription. And then the other thing you see in this slide is that big bar shown in blue. And these are, I'm just going to call this other regulatory sequences. So there's all kinds of other sequences that are in and near the promoter that various proteins can bind to. But the other thing about eukaryotes that addresses that complexity issue is that some regulatory sequences are literally thousands of base pairs away from the coding sequence. So in other words, all the things that regulate gene expression are not right there by the gene. There are some regulatory sequences that are completely in a different place than the gene that they actually regulate. And so, of course, that gives the eukaryotes a lot more space and room to sort of have these nuanced regulatory mechanisms, but it also makes things a little bit more complicated. But there are a lot of other regulatory sequences in addition to the ones that are near the promoter. And I think we'll touch on some of those a little bit. And some of those regulatory sequences are called enhancers. And enhancers are responsible for sort of being able to increase the rate of transcription above that basal regular level. And then there are also silencer sequences that are responsible for decreasing or preventing transcription. So we have the core promoter that just kind of gives you the basic transcription level. And then there are a bunch of other regulatory sequences, some near the gene, some far away from the gene. And some of those regulatory sequences are responsible for being able to increase transcription, and some of those regulatory sequences are responsible for decreasing transcription. So that's the basics of what goes on at the eukaryotic promoter. And just as we talked about with the prokaryotic gene regulation, there are a whole slew of proteins that physically associate and bind to all of these DNA elements that I just mentioned. So there are proteins that bind to that core promoter. And those proteins in eukaryotes are what we refer to as general transcription factors. And in this diagram, they're shown in those kind of tan blobs, and they're termed GTFs, or general transcription factors. And they physically associate with the core promoter. Remember that core promoter includes the TATA box, and it also includes that transcription start site. So there are these generalized proteins that associate there, and they are required for RNA polymerase to come in and begin transcription. But there are also a number of other proteins that bind to some of these other regulatory sequences, some of these activators and repressors that are also important for sort of turning up the dial or turning down the dial on transcription. So let's look at how one of these activator proteins works. So earlier, remember I said there are some sequences that are called enhancers, and so activators bind to enhancer sequences. And you see that in number 1 here, an activator can bind to an enhancer, and that activator then literally enhances the ability of those general transcription factors to come in and bring in the RNA polymerase. So that's how it kind of turns up the dial. It brings and makes this whole process more efficient. So that's the activator side of it. The repressor side of it, there would also be proteins called repressors that bind to silencers that decrease or turn down the general transcription rate of the complex and inhibit the RNA polymerase from coming in. So we've got enhancers and silencers, and we have activators and repressors, and we have general transcription factors. So there's lots and lots of players, some of them at the promoter, some of them far away from the promoter, but all of these things in concert then can work together to turn up and turn down the rate of transcription of particular genes. And again, that adds to that complexity that you see in eukaryotic gene expression. But there's one other important part of eukaryotic gene expression that I want to point out, and this has to do with the actual structure of the DNA. So let's talk about that in the next couple slides. All right, just a quick reminder, you've seen this slide before, and this is just, I want you to pay attention again to the bottom here. So we remember that DNA doesn't exist in the eukaryotic nucleus just as naked DNA. It is tightly wrapped around proteins called histones. And these histone proteins and then and DNA together is what we call chromatin. And certainly if the DNA is very tightly packed, very tightly condensed and tightly wound around these histones, it's not very accessible to any of these proteins that are going to bind to the promoters, the enhancers, the silencers, or anything, right? So the activators and repressors can't access the DNA if it's very tightly packed up. And so there's another mechanism of gene regulation that is something that we call chromatin remodeling. So let's look at a little bit of what that might look like in the next slide. So in this slide, we have various parts of the promoter, the core promoter, and again, the transcriptional termination site are shown here with arrows. But those kind of tan blobs above the DNA, those are histones. And so the DNA is tightly wound around those histones. And what we notice is that the DNA can be in either what we call an open or a closed conformation. And NFR that is indicated here is a nucleosome-free region. So those histone DNA complexes are called nucleosomes. And certainly at the core promoter, the DNA is not wound very tight. It's in an open conformation, or it's loosely packed. And so that allows for the transcription proteins, the general transcription factors, the activators, and repressors to come in and physically bind there. So there are a number of signals in a cell that can change or remodel the chromatin structure. So it can be converted from sort of a closed to an open state. And I'm not going to get into a lot of the details of this, but let's just suffice it to say that when DNA is in its closed or tightly packed configuration, when the chromatin is very tightly packed and there aren't very many of these nucleosome-free regions, it's transcriptionally inactive because the transcription proteins can't access it, can't get to it. And so that's another way that cells can regulate transcription. They regulate the chromatin remodeling. And if it's tightly packed, it's transcriptionally inactive. If it's more open and loosely packed, it's transcriptionally active. So how does that happen? How does that chromatin remodeling actually occur? Well, it's somewhat complicated, but let me just say one other thing about how this works to regulate transcription in eukaryotes. So on this slide, we see some DNA we see some histones here, and you see a number of amino acids in blue, those little blue balls. And you see little M's and little AC's, and we're not going to get caught up in all the details of this. But what we know occurs at genes when they're being regulated. So let's say there's a gene that's being activated. Well, when activators physically bind to the DNA, and histones then get chemically modified, so they get methylated. That's what the M stands for here. Or the proteins on the histone get acetylated. That's what the AC stands for here. What happens then is it signals the remodeling of the chromatin to go from that closed or the inaccessible state to a more open or accessible state. So it's not just about proteins binding to promoters and allowing transcription to occur. It's also about modifying the structure of the DNA so that the promoters and those regions of the genes are actually accessible and available to be bound so that transcription can occur. So we've got all these proteins that bind, we've got all of these modifications to the chromatin structure, and this is happening at every gene individually. And so I hope you can see as we start to combine all these various mechanisms of control that you can have some pretty tight and nuanced control over gene expression in eukaryotes that are just not possible in prokaryotes, because prokaryotic DNA is not packaged into chromatin like eukaryotic DNA is. So that's just a little touch or a little brief outline of some other ways that eukaryotic transcription is regulated. All right, and then just one last thing that I want to talk about with eukaryotic gene regulation, and I'm not going to go into any of the details. One of these we've talked about before. But so far, when we've been talking about both prokaryotes and eukaryotes, we've only been talking about regulation of gene expression at the transcriptional level. Right? Everything has been about how do you increase, decrease transcription, or that synthesis of messenger RNA. Well, we know in eukaryotes we can also affect gene expression by affecting RNA processing. We can affect gene expression certainly by impacting translation, and we also can affect gene expression through process that convert a protein to a functional protein. So I just want to quickly highlight a couple of those things. And again, we're not going to go into any of the details of this. Your textbook goes into a lot more detail, but I'm just going to, what I show here is really the level at which I want you to know and understand it. So let's first talk in eukaryotes. Remember, there's that RNA processing step that you don't have in prokaryotes. So how can that be used to regulate gene expression? We talked about this before with splicing. We talked about this mechanism that's called alternative splicing, right? So remember when you have a messenger RNA or a pre-mRNA that's made, and depending on the cell type, that messenger RNA is being transcribed in, you can have different splicing patterns. And so in this slide, maybe the splicing pattern for a smooth muscle cells gives you the mRNA that's shown on the left, but the splicing pattern in striated muscle cells gives you a different splicing pattern. That is one way to regulate gene expression, right? So you're using alternative splicing to affect how much of a particular protein is present in each of these cell types. So they have different introns, or the introns are spliced out, but they all have different combinations of exons. So alternative splicing is a mechanism by which you can regulate gene expression. And again, this only occurs in eukaryotic cells because splicing doesn't happen to any extent really in prokaryotic cells. So we can regulate at the RNA processing level through alternative splicing. So in your textbook, this diagram is actually fairly complicated, and I don't want you to get caught up in all the complicated parts of this, and so I've only highlighted one bit of this. But this is an example of how in eukaryotic cells translation can be regulated. So let's say transcription occurs just fine. We have messenger RNA present, but what we can do through a number of different mechanisms, and I'm going to show you two examples here, is we can regulate whether or not that messenger RNA actually gets translated. That's another way to affect the amount of protein that's present, right? So this example, again, we're not going to go into all the details, but there's a protein shown here called RISC, R-I-S-C. And it's an RNA binding protein. And so when this protein binds to messenger RNA, it can do one of two things. It can signal that RNA to be degraded. And so clearly, if that whole messenger RNA is degraded, it's not available for translation, and so we don't get protein being made. Or it can block and just inhibit and prevent the ribosome from being able to translate that particular messenger RNA. So this is an example of an RNA binding protein physically associating with a messenger RNA and either affecting the stability of the messenger RNA, in other words, it gets degraded, or blocking its ability to be translated. So this is regulation of translation, right? We're not regulating transcription. The messenger RNA was made, but we are regulating whether or not that mRNA gets translated. And then there's one other example of translation regulation that I just want to show you quickly in the next slide. So again, there's a lot of detail here. I don't want you to get caught up in the detail, but this is another example of an RNA binding protein. The previous slide, we talked about an RNA binding protein. Binds to the messenger RNA, targets it for degradation, or blocks it from being translated. And this is a similar idea here. So the brown protein shown here is again an RNA binding protein. And that RNA binding protein in the top panel there, again, blocks and prevents the ribosome from binding and prevents that messenger RNA then from being translated. So, I mean, when we talked about transcription control, right, we talked about a bunch of proteins that bind to the gene and don't allow transcription to occur. Well, this is an example of a protein that binds to the messenger RNA and doesn't allow translation to occur. Okay, so an RNA binding protein. So this is just another type of example, similar to the one that we showed on the previous slide. But you have proteins that bind to RNA, messenger RNA, and prevent them from being translated. And then the very last thing I want to talk about is we also know, and this is very common in eukaryotic cells, we get genes transcribed into messenger RNA, and then the proteins, or the messenger RNAs get translated into protein. But those proteins have to undergo some sort of post-translational modification in order to be functional. And so this is another way that one can regulate whether or not a protein is active in a cell. So there are some proteins, in order for them to be, and again, I don't want you to memorize everything on here. I'm just showing you a few of these as examples of what I mean when I'm talking about post-translational regulation. So there are some proteins, when they're made right hot off the press, right after they're translated, they actually have to be cleaved. They have to be broken or cut into smaller pieces in order for that protein to be functional. And insulin is a classic example of that. Insulin is made as a big functional or big protein, and then that protein gets cleaved and sort of reorganized before it can function as insulin in the cell. And of course, there are other modifications that occur. Sugars get added, lipids can get added, disulfide bonds have to form, phosphate gets added, acetyl groups get added, methyl groups get added. And so all of these chemical modifications of proteins are another way that we can regulate the activity of a protein in a cell. And this is at the very end of the line, right? After the protein is present. These are post-translational regulation mechanisms.